Hello, everyone. Welcome to this webinar on protein science in South America, brought to you by the Protein Society. My name is Liz Meyring. I'm the president of the Protein Society, and I'm delighted to introduce our webinar today, presenting an exciting range of protein science from all across South America. Before I turn this over to Cesar Ramirez-Sarmiento, who's our leader for the webinar today, I'm going to make just some brief announcements. So I'd like to invite you to join us for our annual symposium this summer in San Francisco. We have a fantastic program, which you can see at our website, and we'll be providing many travel awards. So please keep an eye out for more information on that as well on our website. I also want to highlight our journal, Protein Science, which is led by our outstanding editorial team led by John Currian. I want to encourage all of you to consider publishing your latest research here as well. We have a broad range of protein science across many leading disciplines in the field. And now this brings me to our feature attraction. I want to thank Cesar Ramirez-Sarmiento from Pontificia Universidad Catolica de Chile for his leadership in organizing and chairing this webinar. Some background on Cesar: he obtained his Bachelor of Science in Biology and PhD in Molecular Biology and Neurosciences at the Universidad de Chile, and continued as a postdoctoral researcher there with Jorge Babul and Elizabeth Kamives at University of California, San Diego. He took up a position as assistant professor and was promoted to associate professor in the Institute for Biological and Medical Engineering at Pontificia Universidad Catolica de Chile, where he's investigating metamorphic proteins and plastic degrading enzymes using computational and experimental approaches. He's an executive counselor of the Protein Society, a member of the Global Investigator Network of the European Molecular Biology Organization, EMBO, and the only affiliated PI based in South America of Rosetta Commons, a hub for computational protein design led by recently announced Nobel Prize winner David Baker. Cesar has also been very active in organizing international conferences, and we're very fortunate and grateful that he has put together this wonderful program of talks today. I also want to thank very much our speakers, Andre, Jenny, Lucía, and Ignacio, for sharing your exciting research with us as well. And thanks to our staff at the Protein Society, Raluca Kadar and Shannon Cunniff, for your help behind the scenes, as always. Before I turn this over to Cesar, some brief logistical notes. The way we're going to do this is that each speaker will present for about 23 minutes. Then we'll have a couple of questions for each speaker. And after the 4 talks, we'll have further questions in a discussion format among our speakers and our organizer. So I'm going to encourage you all in the audience to find your Q&A tab where you can type your questions at any time. Only the organizers and the speakers will be able to see the questions until they've been answered. Cesar will be reading out the questions, which may not all be answered in real time, but speakers can also type answers to the questions in the chat, and then they show up in the webinar as well. So I thank all of you in the audience as well for joining us. Your questions are very important, and they really enrich the webinar. So I really encourage you to share your thoughts as well. It really stimulates the discussion and makes everything more engaging. So with that, I'll turn it over to you, Cesar. Thank you all again.
So thank you, Les, for the introduction. So yeah, we're going to have a fantastic webinar with invitees from Argentina, Brazil, and Chile, speaking about the type of computational and experimental protein science that is happening in our area, regardless of sometimes some undesirable funding scheme. Science is really good. And so Instituto de Investigaciones Biotecnológicas from Universidad Nacional de San Martín. Then we're going to have Ambrosio from Instituto de Física de São Carlos at the University of São Paulo. After that, we're going to have Ignacio Sánchez from Universidad de Buenos Aires in Argentina. And lastly, we're going to have Blamey from Fundacion Biociencia in Santiago, Chile. So without further ado, we're going to start first with Lucía Chemes from Argentina. And the title of her presentation is Unexpected Energetics in the Transition State Ensemble for binding of a short linear motif to a folded domain. So, Lucia, the floor is yours.
Thanks, Cesar.
Thank you so much. So with that, we will move on to the next speaker. Thank you so much, Lucia, for your talk. It was extremely fascinating as always. For people that have more questions, please put them in the Q&A. While Lucia is here, she can answer for the incoming questions.
Of course.
So now we're going to have the presentation of Andrea Ambrosio from Instituto de Física de São Carlos from Universidad de São Paulo in Brazil about single particle analysis and in situ cryo-ET, glutaminase filaments as a case study and an emerging opportunity in South America. So Andrea, the floor is yours.
Yep. Hello. Can you hear me, Cesar? That's fine?
Yeah, we can hear you well.
Okay. Perfect. Thank you. Thank you so much, everyone. I'm going to be looking to the screen on my side, so I apologize. I'm going to be facing sideways to the camera. But I'd like to take this opportunity before I start my presentation to express my support to Lucia, speaking about the protest against the dismount of science. So please accept my support in this fight in any way that I can. So, and what I'm going to talk to you about today is from the structural biology aspect of a work that we have been developing in the lab for the past 15 years or so. And this concerns the structural determination, both in vitro and in situ, of proteins that form filaments in the cells. I'm going to contextualize this in a moment. But the title is this, the single particle analysis and in situ cryo-electron tomography of glutaminase filaments. And I hope that this technical approach would also be feasible in South America, as I'm going to show in my last slides. And I'm here, and this is very important to be clear that I'm representing a much, much larger group of people that have contributed in so many different ways to this work, which I'm going to acknowledge in the next slide. But first, I'm going to send my thank you to Cesar, Raluca, Shannon, and all the Protein Society team. Thank you so much for this opportunity. And how can I change my slides? Yep, that's it. So I'm going to start backwards, my presentation, so acknowledging the people involved in this work first. And again, this is only the latest chapter of research that we have been doing in the past 15 years. And this research has been shared in terms of a multidisciplinary approach with Dr. Sandra Diaz, who is in this picture, who only by accident happens to be my wife as well, my longtime partner in science. And the research focus of our groups together is related to the adaptation of metabolism in tumors and how they adapt to environments which are often depleted of nutrients in order to proliferate. These are all the people involved in this manuscript, which I'm showing the title here. The paper was published just about a year ago, October 2023. And I'm very quickly acknowledge people from my group, Sandra's group, the collaborators that helped us with the cryo-EM experiments in single particle analysis, the people from EMBL Heidelberg from cryo-electron tomography, and Silvio with some microscopy also at Unicamp. And this presentation actually, again, is only a small chapter of the research. And in terms of structural biology itself, the research started in 2009 when I started my laboratory. I've been trained as a protein crystallographer. We answered at the moment as many questions as we could in terms of the molecular mechanisms of glutaminases in the past. And then crystallography only proved to be useful for certain cases in where you don't have protein aggregation, which makes the sample untreatable by crystallization. And then there is a hiatus in this research for about 5 years or so when we had access, where the technology developed as well, and we had access to cryo-electron microscopy. So the work is divided in 2 stages, or into time slots, first by X-ray crystallography, and then more recently by cryo-EM. And again, I'm only showing the structural part, but there is the work is complemented by extensive cell molecular biology, biochemistry, and biophysics. And the key word for us throughout this process is integration. We're very proud on how we can integrate all these different approaches to try and generate, hopefully, a more complete or holistic understanding of how the proteins of interest work. And in this case, glutaminase. So specifically, the cryo-EM will relate to the filamentation state of this protein glutaminase. And there is a whole deal of experiments that try to show how this filament is important to protect the cells from mitophagy, the cancer cells from mitophagy, but I will not touch basically in any ways upon this aspect of the research. And this part of integration is very important for us as well. And so that's what we've been trying to achieve across the years. And the paper that we published that is being shown in the previous slide, this paper was acknowledged as an example of integration in the editorials that celebrated the 30 years of the journal Nature, Structure, and Molecular Biology last year. So the previous and the past and the current editors of this journal, each one selected a manuscript that they wanted to highlight in any way they thought important. And the editor, Katarina Czesinska, she has been an editor since 2022. She selected our manuscript specifically in this aspect, how we can integrate different experiments in cell biology, molecular biology, cryo-EM, single particle crystallography, and tomography to provide a mechanism at all scales on how this protein could work. So this was the greatest honor for us. I'm going to just skip briefly. And before I move on to the structural aspect of this, I'm just going to provide a very brief context on where the biology of this research is at. And the key point for us is tumor metabolism. And this slide, the picture here at the bottom, it highlights in very general, in a very general way, the processes that the cancer develops, going from a mutated cell that acquires an oncogenic property, becoming a cancer itself, and then growing in terms of the size of the tumor. This tumor eventually becomes very large, such that it can invade the adjacent blood cells, and then eventually metastasize to somewhere else in the body. That's which is usually very undesirable. And in this process here, you can see that the number of cells often increase rapidly and in a very chaotic way. And this is extremely important for the tumor to be able to grow in this way, but it has to be supported by a hallmark, which is a very important feature, which is related to how the cancers, they deregulate the cellular energetics and the biosynthesis, biosynthetic processes as well, in order to be able to reproduce very quickly. So the regulation of the cellular energetics is a hallmark, and it's known as the Warburg effect, which was first described by the physiologist Otto Warburg about 100 years ago. And the reason behind this such important feature is extremely simple. The tissues, the cancerous tissues, they often grow in an abnormal and in excessive ways, meaning that they have a very highly proliferative phenotype. And this, of course, has an increased demand for energy production and the building of biosynthesis or biosynthetic blocks itself. So the cells must adapt the metabolism, and quite often the environment is very poor in terms of supplying nutrients for this purpose, for metabolic purposes. In particular, the glutaminase proteins, they're involved in the metabolism of glutamine, the glutaminolytic pathway, such that glucose and glutamine are typically the most important nutrients for particularly for cancer cells, which are in the top here. And the glutamine is metabolized inside the mitochondria in this context. And the key step, and the first step in this glutaminylated pathway is the conversion of glutamine into alpha-ketoglutarate, which is done by the glutaminase enzymes, which were first described by Hans Krebs back in 1932. And so what is important for the rest of the talk that I'm going to talk about is there are two key aspects that I want to briefly touch upon. The first is the molecular mechanism of glutaminase activation. So up until 2013, we could show that this glutaminase protein would become active towards the substrate in shifting its oligomeric state from inactive dimers to active tetramers, and in a way that was important or dependent on the presence of inorganic phosphate in the medium. Even though phosphate does not necessarily directly participate in the catalysis, it does have an allosteric effect in the tetramer, making the tetramer becoming active. So about 10 years ago or so, we could understand this aspect of the activation mechanism of the protein. And then as cryo-EM became accessible, we could understand that this protein, the tetramer, was not the boundary in terms of size, but that the protein could extend into more much larger and heterogeneous filaments. And so the shift between the oligomeric states of the protein is extremely important, but also it is important to have in mind that the protein sits in the mitochondria, and the mitochondria itself is very dynamic in terms of the morphology. And usually what we call the normal state of the mitochondria depends on the homeostasis of the cells. But if you apply some sort of stress, you can either make the mitochondria very large, forming tubules, or make it very small in an efficient way that becomes much spread across the cells. And in particular, starvation of nutrients. And it's been known as from a long time that it promotes the elongation of the mitochondria. And this is the key aspect that I'm going to touch upon that will be linked to the filamentation of glutaminase. So I'm showing here just a summary of all the cell biology experiments that have been done for this work. And I hope you can see the time lapse here showing the transition in the colors that are applied to this image upon glutamine depletion. So we cultivate the cancer cells that have been stained for glutaminase in green and for mitochondria in red. And you can see, I hope you can appreciate, as the time goes by, the signal in green becomes much more discrete and elongated. And then when there's yellow on top of it, meaning that there is also a mitochondria that's undergoing the same sort of processes. This is the beginning. You see, you have a very clear yellow overlay of the two colors. And as the time goes by, glutaminases start fragmenting. So does the mitochondria. And then the mitochondria elongates along with the glutaminase to form this hair-like structures. The hair-like structures, I guess, are more easily observed in the panel, on the right side panel, where on top we stain for mitochondria, glutaminase, and then there's the overlay. This on top is the presence of normal amounts of glutamine, meaning that they're what we call the normal mitochondria size distribution. And then at the bottom we basically deplete glutamine, and then the mitochondria becomes tubulated and the glutaminase becomes elongated, and suggesting the formation of filaments. And then when we overlay, we have this spike or hair-like structures. And this phenomenon is only dependent on glutamine itself, meaning that if we remove glucose, which is another substrate that is important for the cancer cells, you don't have this different distribution of glutaminase protein, only in the case of glutamine. And this is reversible. This phenotype here is reversible depending on whether we have we add the subsequent nutrients or the substrates in the glutaminolytic pathway, or if we add an allosteric inhibitor of glutaminase itself. Cesar, I'm going to ask you to keep track of my time because I'm afraid I could speak much longer than 25 minutes.
That's good.
And then, these are only an example experiments in this case, showing the phenotype that I want to move forward in my presentation, but there's hundreds and hundreds of experiments regarding this, the proofs and the counter-proofs of glutaminase filamentation and its involvement in mitophagy protection as well. But the bottom line is that under glutamine deprivation, we have the formation of elongated mitochondria. And this elongated mitochondria contains glutaminase, which is condensed, and these condensed state protects against mitophagy. So we wanted to understand what this elongated state of glutaminase meant. So we performed electron microscopy experiments because, again, since the protein is forming filaments, these filaments, they're extended and heterogeneous in size. So these are often completely non-treatable by crystallography because the crystals require some homogeneous and distributed populations of proteins. So we went to cryo-EM. This is a negative staining image of the protein, so in the presence of phosphate. So I hope you can appreciate filament-like structures here. And then, but negative staining does not allow you to solve the structure at high resolution. So then we moved to cryo-EM. Upon setting the grids under vitreous ice conditions, most of the filaments, they disassemble for whatever reason, we can discuss, but we have some elongated states that are kept. I hope you can see them here. I think I have, yes, there are arrows pointing to these elongated states, which across the entire data set, they were enough for us to isolate and to solve the structure using a single particle picking approach and then a helical refinement to reconstruct the volume. And these are representative two-dimensional classification of the particles that we picked. And then we managed to reconstruct a volume for the filament according to a resolution threshold given by the Fourier-Shell correlation as 0.143. Giving us the highest resolution or the global resolution for the data set in the range of 3.1 angstroms. And this was a map that allowed us to determine many of the features that were previously unknown for us. In order to describe the filamentous state of the protein, we could see side chains, we could see the phosphates, we could see everything that was important. And so I'm going to make this story very short. At the bottom here, this is the filament. This is the volume of the filament, showing that the filament is helical in nature. So we have 7 angstrom step in the across the helix as each tetramer of glutaminase associates side by side, and the colors, the like colors, they represent the same subunits being transition from across the helical axis. And then every time we incorporate a new tetramer, there is a rotation of 48 degrees, meaning that there's about 6 or 7 tetramer units in order to make a complete turn of the helix. And what is important for us in terms of the mechanism is to isolate 2 tetramers which interact side by side. So here we have 1 tetramer, the lateral association of the other tetramer. And then I want to highlight here the interfaces, the key interfaces for us, which are the dimer interfaces. Remember in the beginning, I mentioned that the dimers of glutaminase associate into tetramers. So now we have the dimer state, the dimer interface within the filament. We have the tetramer interface within the filament, and we also have now the filament interface between the tetramers. All very new interactions for us that are very much different from the crystallographic states that we determined about 10 years earlier. And I hope this information is better synthesized if I show you the transition between the crystallographic state and the filamentous states for the tetramer. And I hope you can appreciate the amount of conformation changes that you can get, especially within the central part of the tetramers, which are related to the active sites of the protein. So there's a whole lot of transitions occurring, especially in a loop that we call the activation loop of the protein. And this is very important. Apologies. The movements are very important for describing the molecular mechanism of the tetramers, but this is not, again, what I'm going to touch upon today. The thing is that I want to go to the in situ structure of this protein. So in order to prove that the filament that we observed in vitro, in this case by single particle analysis, was also perhaps related to the elongated state or the condensed state that we're observing in cell biology. So then we took the cells that were making the phenotype here at the top, as I showed you in an earlier slide, and we moved to cryo-electron tomography studies by combining the tomography experiments with the lamella production. And this was done by cultivating the cells of interest on top of electron microscopy grids. And then when we cultivate the cells there and they're labeled with some fluorescent tag, I'm going to show you why this fluorescent tag is important. We freeze the cells on top of the grids. And since the frozen samples containing the cells and the buffer surrounding the cells are very thick so that the electron cannot go through the entire samples when doing a transmission study, we can use a microscope, which is called the dual beam microscope, which performs the fabrication of thin lamella within the samples by destroying everything that surrounds the region of interest by using a beam of focused ion beam of gallium ions. So we identify at which point of the cells by light microscopy where the protein, the regions of interest are, and then we destroy the surroundings, only leaving a very thin sheet of intact samples to perform tomographic studies. And this is one example of the experiments that were done. This, I hope you can see here, this is the boundaries of the squares in a cryo-electron microscopy grid. We have this drop, what seems to be a drop here, which is a scanning electron microscopy image of a cell that's surrounded by a layer of liquid. And then if you superpose with the fluorescent tag, you can see that it labels for the GFP tag that is bound to our glutaminase protein. This is a low resolution experiment because in this case, we do not require high resolution fluorescent microscopy, but we do get high resolution electron microscopy. So from this cell that has the fluorescent signal, we destroy the surroundings, we identify the regions of interest, and then we destroy the surroundings so that we end up with a very, very thin layer of about 200 nanometers in thickness for the intact cells. And then we perform transmission microscopy analysis. And as the zoom goes by, hopefully in the previous picture here, I hope you can see on the screen that we can see some organelles. And there is a very large mitochondria spanning this region where the red cross is. There are other mitochondria and some other organelles here, but we end up collecting a high resolution tomographic data set or a tilt series that allows us to reconstruct the entire volume, which is underneath this cells. Let me just, I'd like to start my movie here. How can I start my movie? Yeah, so this is the movie. This is the tomographic reconstruction of the lamella that was collected by transmission electron microscopy. And then I label here some organelles that can be very easily identified. And of specific interest for us is a mitochondria that is going from this side of the screen all the way to the top up here. And I hope you can appreciate that within the boundaries of this mitochondria, and you can see the double, the bilayer here, the double membrane system of the mitochondria, there are a stream-like pattern of something that look like filaments traversing the entire organelle. I'd like to compare this to a stream of water going down the river. I hope you can see this from your screen because this for me is one of the most beautiful results that I have ever seen, especially working with this protein for many, many years. And so, of course, we had done all sorts of controls that we could, showing that this strict or this stream-like phenotype was related to the depletion of glutamine itself. This was happening both for the GFP tag glutaminase and for the endogenous protein itself, label-free, and also not happening in the mitochondria when we have normal amounts of glutamine. And this experiment on the right was done a couple of times. And then from the identification or the automated identification of filament-like structures by softwares, available softwares, we could identify filaments. And these filaments were chopped into very small pieces and then superposed or averaged to provide what we would hope was a higher resolution representation of the smallest repeating unit. And this was done, the reconstruction was done at a 28 angstrom global resolution, maximum resolution. And this here is the, in gray, you have the structure, the low resolution structure that is averaged for all the filaments, for thousands and thousands of filament particles chopped down from the top image here. And hope you can see that this the gray structure, the gray envelope is filled with the high resolution single particle structure that we ended up obtaining by the in vitro structure. So there is a very, very high correlation in terms of the geometric parameters from the structure obtained from the inside the mitochondria to the structure obtained by the in vitro, especially the dimensions of the helix and the rotation and the angles that are necessary to describe the geometry of the helix. And then here you have a cross section showing how well our low resolution structure fits the in situ envelope.
Okay, Andre, time to wrap up.
Yeah, just one more slide.
Yeah.
Just
Cool.
gonna wrap up. Yeah. So this is the volume reconstruction, the segmentation of the tomographic volume that are reconstructed, that we reconstructed in the previous slide. So here you can see some organelles. We can see some boundaries, some double layers. You can see some microtubules. You can see lots and lots of ribosomes surrounding our mitochondria of interest, and then the glutaminase filaments colored in the rainbow like color palette in this case. And this is the, hold on, the structure that is superposed from the two experiments. So just to finish my presentation, I'm just going to touch upon how we can try, hopefully, to perform these experiments in South America. And that's what we've been working very tirelessly now, is to hopefully have these experiments accessible to us. So there are many ways in which you can perform single particle analysis. You have all sorts of flavors of transmission electron microscopes. And then in order to do the in situ structure, you need to fabricate the lamella. And there are also some equipments that are available from companies across. And then of course, you also need the sample vitrification in order to freeze your samples. And if you look, even though there are lots and lots of equipments available for this, if you look at the global distribution, I'm going to skip this EDB data bank here because it's not relevant for what I'm talking next. But I want to end up my presentation by showing you the global distribution of cryo-electron microscopes around the globe. And I gathered this information this morning, so I hope this is very accurate. On top are the microscopes that are necessary to collect the transmission like experiments. And then at the bottom, the microscopes that are available for fabricating the lamella. And I hope you can appreciate the global distribution is completely skewed to the north part of the hemisphere, or the globe to the northern hemisphere, and it correlates very strongly to the economic development of the region, where you have like almost 800 high-resolution microscopes in the globe. Pretty much 95 or 98% of these microscopes sitting in the northern hemisphere, only a handful in Australia and in Brazil. And there's a similar scenario for the lamella fabrication microscopes as well, where basically 99% of these microscopes are only available across Europe, the United States, or East Asia. So this here is to show to you that you may have access to experiments soon to do this type of experiment, particularly in order to collect high-resolution transmission electron microscopy to solve the structures at our institute in São Carlos, at Federal University of Rio de Janeiro, and Helena Campinas, and also to fabricate lamella. We're finishing installation of our dual beam microscope in São Carlos, which is only a partner of the Rio microscope as well. There are only 2 microscopes of these available at the moment in South America. So our facility, and this is a scenario that's very similar for crystallography, because in order to solve the structure, you need a very crystal structure, you need a very good X-ray beam, and the distribution of synchrotron sources is pretty much the same for the electron microscopes that I showed you earlier. Campinas has the Sirius fourth generation synchrotron source. There's only 2 in the southern hemisphere of the globe. So this is my last slide, I promise. We're setting up this facility, hoping to provide as a multi-user facility open to the entire community in South America or anywhere else, but from where people want to come from. For the past 2 years, we applied for a grant to acquire these microscopes in 2022. We've been granted in August 2023, and then we started installation and site remodeling in order to getting this working. And then the microscopes, they're entered commissioning 2 months ago, and we hope to have community access by proposal submission in July. And these are the 2 pictures of the microscopes. This is a transmission electron microscope Tundra, and then this is the Atlas for lamella fabrication. So I'm gonna end here, and I wanna thank you all for your attention and for the opportunity as well to present here. And I hope to see you soon collecting data at our facility in São Carlos. Thank you so much, Cesar.
Great. Thank you, Andre, for the presentation. That was pretty cool. And it's pretty wonderful to see how all of the instruments that Brazil is getting for doing this type of measurements and technologies. This is pretty amazing.
Thank you.
So not to affect the schedule that we have, we're gonna ask for the people to ask questions in the Q&A, but we're gonna move on to the next presentation so that we don't run short of time. Andre, just keep looking at the Q&A. I'll let you know if there's some incoming questions for you. Awesome.
Cesar, have I stopped sharing my screen?
Yeah, you have. Yeah.
Oh, okay. Thank you.
So we're all good. So now we're going to move on to the presentation of Ignacio Sánchez from Universidad de Buenos Aires in Buenos Aires, Argentina. And he will speak about solvent constraints for biopolymer folding and evolution in extraterrestrial environments. So Ignacio, the floor is yours.
So thank you, Cesar, and the organizers for this opportunity. Thank you to everyone who's come to listen. I'm going to present some work we did in the recent past, together with Ezequiel Galpern, a postdoctoral student in our lab, and Diego Ferreiro, the co-director of the Protein Physiology Lab. We've done this at Universidad de Buenos Aires and CONICET, with some funding from the NASA Astrobiology Institute. Let's say we are at home at night and think of what's out there. Close by, there's the sun, there are some planets. Moving on further away, there's our galaxy, the Milky Way. That's 10 to the 11 stars. Potentially, that's a lot of planets. Even further away, there are 10 to the 11 galaxies. That makes up for 10 to the 22 potential solar systems. The universe is huge. Wouldn't it be surprising that our planet is the only one with life? We think it would be really surprising. We expect life to be out there. Next question that comes to mind is, if there's life, how does it look like? Is it like in movies, like some human-like bipeds that can communicate with us? Is it something different? We don't know. We are a protein lab, so we look at it from a molecular viewpoint. So we say, if there's life, there must be biochemistry. We postulate that life works with liquid solvents and acts through biopolymers. About solvents, we know life in water, but it's been proposed before that some other solvents may give support to life in extraterrestrial environments. Say ammonia, formamide, liquid nitrogen, carbon dioxide, hydrogen cyanide, even hydrazine. Lots of them have been proposed. Our specific question is if biomolecules, as we know them, can occur in those solvents. And first, let me explain how we conceive biomolecules. They are biopolymers, so they have a sequence. These sequences in some environment, including solvent, give rise through the process of folding to structures. That takes place in microseconds to seconds. Structures move, there's dynamics, and these dynamics give rise to biochemical activities that may take place in milliseconds to years. These biochemical activities contribute in some way or another to organism fitness, that is, they partake in some function. And closing the cycle and back to sequences, the contribution of a folded and moving protein in an organism to its fitness leads to some evolution processes that feed back to the sequences. So sequences fold, function, and as a result of their folding and function, evolve in a much slower time scale. To sum up, biopolymers, in order to function as we know them, must fold and evolve. We look at it from a very simple theoretical way. If biopolymers need to fold and evolve, then some very simple theories such as energy landscape theory and molecular information theory should apply to any functional biopolymer. We can better discuss those theories through the characteristic temperatures for biopolymers. I'm going to walk you through this equation. From right to left, there is the folding temperature, the temperature at which the biopolymer unfolds. If the polymer is to function, the folding temperature must be higher than the physiological temperature at which the biopolymer works. Energy landscape theory further tells us that both the physiological temperature and the folding temperature should be higher than the glass temperature of the polymer. The glass temperature of the polymer is the temperature at which the polymer ceases to explore the conformational plane in an efficient manner. It still moves, but in a very, very slow and inefficient way, which does not allow for folding or function. Last, from energy landscape theory, the glass temperature of the biopolymer should be higher than the pseudo-temperature at which sequences can be described to evolve. So, if a biopolymer is to function, the folding temperature should be higher than the glass temperature. If it is to evolve, the glass temperature should be higher than the selection temperature. These two theories I mentioned, energy landscape theory and molecular information theory, establish from first principles relationships between these temperatures. For example, energy landscape theory gives us this relatively simple equation to relate the folding temperature, the selection temperature, and the glass temperature of the polymer. In turn, molecular information theory defines some efficiency for the conversion of free energy of folding to information in protein sequences. That efficiency, it follows from theory, equals the ratio between the selection temperature and the physiological temperature. We can simplify this to the ratio between the selection temperature and the folding temperature. This has an upper limit of around 0.7 or the natural logarithm of 2. And that's all we need. From this linkage between the characteristic temperatures for folding and evolution, we can describe that there's a compromise between foldability and evolvability of a biopolymer, which is independent of solvent and biopolymer. For a biopolymer to fold and evolve, according to energy landscape theory, the ratio between folding temperature and glass temperature should be higher than 1, and also the same stands for the ratio between the glass temperature of the polymer and the selection temperature. Information theory refines those limits and puts them a little higher, so that the biopolymer is not only evolvable and foldable, but also codable. Let's go slowly towards specific examples. First, we can discuss the biopolymers we know, that is, proteins, DNA, and RNA, in the solvent we know for life, which is water. We know water boils at 373 degrees Kelvin. It freezes at 273 degrees, but other things could happen. Above the boiling point, there can be superheated liquid water, which is metastable, up until the superheat limit. Below the freezing point, there may be supercooled water, down to the supercool limit at 231 Kelvin. Below that, there are two transitions. One is called the glass transition of water at 136 Kelvin, and the other one is the one that will interest us today. That's the dynamic transition at approximately 220 Kelvin. The dynamic transition of water is the one where solutes stop moving efficiently in the solvent. That's the left part of the figure. The right part considers terrestrial biopolymers. Typically, in our labs, we measure the folding temperature, we know the physiological temperature, but also a few experiments have characterized the glass transition of biopolymers. That's the blue circles, squares, and triangles for proteins, DNA, and RNA, which show that on average, the glass transition of biopolymers is around 207 Kelvin. That is, it corresponds to, within the experimental error, to the dynamic transition of water. This makes sense. In order to fold, biopolymers should move in the context of a solvent. They cannot do it faster than the solvent. Therefore, it makes sense that the glass transition of the polymer will evolve down to the limit of the dynamic transition of water. And as a consequence, as Hans Frauensfelder described, the polymer motions will be slaved to water dynamics. To keep in mind, the glass transition of biopolymers corresponds to the dynamic transition of the solvent. How can we consider different solvents and evolve folding and evolution? In any solvent, we will know the freezing and boiling point, and we can postulate that the folding temperature can take values between these two, and that the glass temperature of the biopolymer will evolve to converge with the dynamic transition of the solvent. In that case, we can use the equations from energy landscape theory and molecular information theory to calculate conditions where folding and evolution can happen. That can take place if the ratio between the folding temperature and the glass temperature is higher than 1.373 approximately, and the ratio between the glass temperature and the selection temperature is higher than 1.048. And if we know that, we can also calculate the value of the efficiency for conversion of folding free energy to information. That's the theoretical framework. Next, we got our hands dirty and worked with a dataset of solvents and our equations. Our solvents were water, 10 different hydrocarbons, 19 small substances of low molecular weight made of elements that are abundant in the universe, that is carbon, hydrogen, nitrogen, oxygen, and sulfur, and 24 aromatic and halogenated compounds. In order to apply our theory, we need to know the dynamic transition temperature of each solvent. First problem, we didn't know that for the 54 solvents in our database. So we estimated it. Our estimation comes from the well-known linear relationships between characteristic temperatures of substances. For example, and that's the graphic to the left, the melting point of the solvents we consider shows a fairly good linear correlation with the glass transition temperature of the solvent. The same is true for the boiling point. Finally, that's the plot to the right. We had data for a few of them, and it would seem that there's also linear correlation between the dynamic transition temperature, the one we want to use, and the glass transition temperature of each solvent. Having established this correlation, we use the known glass transition temperatures and use them to estimate the dynamic transition temperatures for all solvents in our database. With that, and the melting and boiling points, we can evaluate each solvent. For example, top graph, that's the ratio between the folding temperature and the glass temperature for all our solvents in our database, in each case for the temperature range where the solvent is a liquid. The thick black line is water. For most of the liquid range, it is above the theoretical limit designated by the blue line. In the case of other solvents, all of it may be above the theoretical limit, only half of it, or only a small fraction of the liquid range. So solvents differ in how they fulfill this condition in their liquid ranges. We can do the same for the efficiency, checking that it's below the theoretical upper limit, and the ratio between the glass temperature and the selection temperature, checking that is above the theoretical limit. The main conclusion here for all 54 solvents in our database is that all of them present a range in which biopolymers could exist. That is, they could fold and they could evolve. So many solvents are in principle suitable for life. Having said that, we tried to rank them. We defined a biopolymer solvent score. That's the geometric average between two things that seemed interesting to us. First, the normalized efficiency for conversion of folding free energy to sequence information. Second, the fraction of the liquid range that is apt for biopolymer folding and evolution. That's the formula on top. In the plot to the left, you have one term in the x-axis and the other term in the y-axis. A perfect solvent with a score of 1 would be at the upper right corner. No solvent is up there, but as I said before, all solvents are in the plot, are in some way or another apt. Are they all the same? No, they are not. The ranking of the biopolymer solvent score is to the right. You see that there's a wide range of scores from approximately 0.9 to approximately 0.4, and that water is close to the top. So all solvents we consider can lead to biopolymer folding and evolution, but water seems to be particularly good. You may argue that these properties of solvents, the characteristic temperatures for freezing, boiling, and the dynamic transition, would vary as a function of pressure. That may be so. The question is how much. We used water to consider this and calculated the biopolymer solvent score of water, that's the black line, in the range of pressures where we find water in our solar system. And we find that even though the components of the score, the purple and orange lines, do vary significantly within this range, the effect of pressure is not so strong above 0.03 atmospheres. So is there going to be an effect of pressure on our calculations? Yes, but not a very strong one. Summing up, in our solvents, the glass transition temperature of the polymer can be approximated to the dynamical transition of the solvent, as we see for biopolymers in water. Water seems to be especially good for biopolymer folding and evolution, and it's also abundant in the universe, so it makes sense that we are water-based life. However, many other solvents are compatible with the folding and evolution of some biopolymer. We postulated in the beginning, if there's life elsewhere, it should have proteins. We walked one step towards proving that it may actually have something similar to proteins. The next questions could be, for example, if we can expect them to be always made of 20 alpha L amino acids or not. And coming from that, what do we actually think is a protein? We think a minimal useful definition is a foldable, codable, evolvable polymer that may be present in many places in our universe. These results with the details of the calculations were published last year in PNAS. Having said NASA to our press agency, this had quite an impact on news, and my favorite is this one, speaking of a relentless search of some humanoid by NASA and CONICET. Quite different from the actual work, as you see. Discussing science with society is sometimes complex, and we are not trained for it, so we better listen to the experts to avoid this kind of situations. And we better talk to society because, as Lucía very well explained with all the details, right now, Argentine science and US science are under attack. And I must say, we are 1 year ahead of the US, and it does not get any better. So what can we do? Should we pretend everything's fine and go on doing science? Well, by all means, we should go on doing science, because if humanity is to persist for a long time, we need science and we need technology. And this should be a glitch. But we should also get out of our labs a bit and defend what we think is worth doing. And perhaps one way to start is to get together and chat. So thanks everyone for your attention and the organizers again for this opportunity.
Awesome. Thank you so much, Ignacio, for the talk. That was pretty impressive. Also fascinating that when people mention NASA, they think about ETs. So I do understand the perhaps frustration of having an image of an alien showing up instead of a biopolymer in the news. So we have one question from Christian Wilson, which is, for proteins, experimentally the glass transition temperature is obtained because at 200 K, the water buffer solvent will be frozen, or is this an extrapolation? And how can you differentiate wrong falling pathways with glass transition? It's kind of very intertwined question, but hopefully you could understand.
The glass transition temperature of biopolymers has been determined for not only proteins, but also folded DNA and RNA molecules. This is usually done in very thin metastable layers of water at that very low temperature. That very thin layer of water can stay liquid for long enough to make measurements of how the protein moves. We may also do that by performing molecular dynamics simulations at very low temperatures, where we just do not allow our solvent to freeze.
Okay. And then a question from myself. I've seen several works showing potential minimal codes for biopolymers in the origins of life when we think about water. Do you think something similar could happen when we're thinking about other solvents and the foldability of such biopolymers, that there has to be a minimal code for these biopolymers at all?
Our thoughts on this are actually under review right now. The headline, in our opinion, would be that there's a minimal size for the biopolymer alphabet, and that the minimal size is determined by the conformational degrees of freedom of the biopolymer. So in a very brief manner, if the biopolymer has 5 degrees of freedom per bond, then the alphabet should be at least of 5.
Right. That's a pretty brief and excellent question. I'm very excited that is under review, so we will know more about it in the future. All right, so thank you so much, Ignacio, for the presentation and being in consideration with time. We were gonna grab up here. There are a few questions, if you don't mind, Jenny, that are on the Q&A that you can take a look at if you want to respond. But for now, we will like to thank Lucía, Andre, Ignacio, and Jenny for the presentations in this webinar. Hopefully, this is not the only webinar about protein science in South America that we will have, sponsored by the Protein Society. Hopefully, in the future, we will have another one with other set of speakers. But this is just to showcase that the science that is happening in our region is pretty cool. And as Lucía and Ignacio mentioned, there's an urgency for supporting our friends in Argentina regarding science and supporting their defense for science in Argentina. So that was a pretty commendable effort from them to put it in front of this webinar. So with that, yeah, thank you so much to everyone to participate in this webinar. We will hang out for a little bit if you have any conversations or questions that you might want to ask. I think that in this case, Jenny's already like typing some answers for the questions that were left off, but the floor is open for any type of discussion that you want to have. So Lucía, Ignacio, Andrea, I don't know if you want to add anything else. So you're more than welcome to do so.
If I may, I have a couple of questions.
Go for it.
Andre, one hypothesis that comes to mind is that filament formation by your transglutaminase is the physical cause for the elongation of the mitochondrion. Have you compared the free energy that is released in the polymerization to the deformability of the mitochondrial membranes or something of the sort?
Can you hear me? Okay, good. Well, thanks for the question, Nacho. A beautiful presentation, yours, very interesting as well. I do not know the answer to your question. To be honest, I have no idea what the effects of these filaments are in the mitochondria at this moment. We have the hypothesis that this could be some sort of a skeleton structure, like the mitoskeleton, but this is too early to say anything in this regard because we still haven't done any experiments. We have plans of performing experiments with point mutants that remove the catalytic ability of this protein, removes a response to phosphate that promotes filamentation, regardless of other effects. But these are still all undergoing. I have no idea at this moment. And the cell biology to this has to be coupled as well with the proteins that constrain the mitochondria in order to, it's like a battle between of forces between the proteins that want chop down the mitochondria versus the mitochondria that has the filaments inside. So this is an excellent question, and I have no idea how to answer this at this moment.
Looking forward to your next seminar.
Thank you. Thank you so much.
And Lucía, if you allow me, I find amusing how science works. You see, first the community studied protein folding and rationalized fast folding together with evolved smooth energy landscapes that allow for fast exploration of conformations and therefore folding in reasonable biological times. And the key tool for that were 5 values. Now we started applying 5 values to protein motif or protein peptide interactions. And as you very well described, those energy landscapes are not quite so smooth. There's roughness almost every time you look at it in detail. So it would seem they have evolved for something else. And I find that intriguing. What do you think?
Well, yeah, I think it's intriguing as well. So the thought that comes to my mind in with the findings that we have is that it's actually barely stable to form these interactions, and that it may have to do with how dynamic these interactions have to be. For me, the most surprising part of the results is that the residues that make the most difference for, let's say, a viral motif to be able to hijack are the same ones that make it almost impossible to cross the barrier if you didn't have the electrostatic interactions helping. So what I think is that actually these interactions are unlikely to happen, to form, and it's made me view electrostatics as a very important force, which we knew it's important for crossing these barriers, but it seems to make absolutely all the difference. We don't have that big contribution from the hydrophobic effect so much. And I find it intriguing that everything that's interesting from the light of the evolution is actually frustrating the transition state instead of making things easier. And the other option, I guess, is I think we were commenting a while ago, that also the IDPs mediate other processes like phase separation. So they may have they need to have these very dynamic ensembles that are completely different in terms of thermodynamics to the folded domains. But I think we're starting just to scratch the surface of understanding their energetics. We don't really. I had one comment that I briefly commented to Nacho after he was asking what are proteins and, you know, what happens with the chirality of the amino acids. And I just commented that there's a recent, very interesting work by Berthe Kragelund actually showing that for IDPs, you can replace L and D amino acids with almost no penalty for interactions. And the restriction to chirality appears as you need to have more order in these interactions. So I found it interesting that these IDPs and globular domains seem to be a little bit extremes in what they allow and what they restrict in terms of protein evolution, something to think about.
They open our minds.
Yeah.
I think Jenny also has a comment or question. You have to open the microphone, Jenny.
I have a question to Ignacio. when he was talking about the minimum size of the polymers of the amino acid structures to consider proteins, he mentioned something about 5 or 6 amino acids.
Not as the length of the polymer, but at the size of the alphabet.
Okay, and minimum lengths of the polymer?
We did not work on that specifically. For globules, it's around 30 or so.
Yeah.
And there's
Well,
also maximum.
I was paying a lot of attention because I am working with amyloids, and I have detected that several small sequences of amino acids could have catalytic activity. And because you were searching for life or potential life or conditions to have life somewhere else, I just want to mention to you that we have been testing peptides of up to 12 amino acids, which we have tested lacase activity, we have tested lipase activity, we have tested RNA polymerase activity. So,
well, that's exciting.
So it called my attention, your presentation, and maybe it could have a connection or could be interesting to you.
Yeah, I think the bottom line of perhaps the 4 seminars is that we need to go beyond the single globular domain paradigm.
Yeah.
So we need to consider larger things, smaller things, liquid-liquid, phase separation, intrinsic disorder. There's a lot of work to do.
Yeah, and remember that, well, I don't know if you, the amyloids are extremely resistant. So, and we have even tested with like oxidoreductases that the amyloids have catalytic activity. Yeah.
Well,
it could be sort of a small version of what we consider a real protein, a real enzyme, you know?
Yeah, redefining proteins.
Yeah, probably. Don't know. We are working on it. Yeah.
Hello there.
Yeah, we have a visitor.
He maybe has an opinion. We don't know.
Yeah. Yeah. Maybe the opinion is that we have to wrap up. So thank you so much again to the speakers for this webinar. We hope to repeat another webinar soon about how protein science is doing in South America. Hopefully better for Argentina. As we know, there are some issues that have to be solved with extreme urgency. With that, just a few reminders. Protein Society is going to have an annual symposium coming in San Francisco in the middle of this year. And there are travel awards with the deadline tomorrow. So if you plan to attend, try to get one of the travel awards. Information is on the Protein Society website. And with that, thank you so much, everyone. And I'll see you next time.
Thanks so much, Cesar, for the initiative. It's really great.
Thank you, everybody.
Thank you.
Bye. Thank you very much.
Bye-bye.
Bye.
Bye.
Bye-bye.